Chapter 21 is about blood vessels and circulation. And you can see from our man here who has had all of his tissues removed except for the blood vessels. The blood vessels are everywhere in the body. So just remember that blood goes everywhere all the time in the body. The blood vessels are classified by their size, their histological organization, and they are instrumental in overall cardiovascular regulations. The largest blood vessels attach to the heart, and these include the pulmonary trunk, which carries blood from the right ventricle into the pulmonary circulation, and then the aorta, which carries blood from the left ventricle to the systemic circulation. And the largest veins would be the inferior and superior vena cava. So remember that arteries carry blood away from the heart. Arterioles are the smallest branch of arteries that lead directly to the capillary beds, and the capillary beds are the smallest blood vessels with thin walls, and they are the location of exchange between the blood and the interstitial fluid. Now, not on the slide, we have venules, which capillaries feed into, and then venules feed into veins. So the arteries get smaller and smaller as they go away from the heart, and the veins get larger and larger as they return to the heart. So the veins carry blood to the right atrium. The blood vessels have three layers. The inner layer is the tunica intima, then we have the tunica media in the middle, and the tunica externa on the outside. Tunic means coat. And we can look at those structures on this diagram. So on the left side we have an artery, and over here we can see an artery and a vein, and you'll notice the difference. The tunica intima is made out of endothelial tissues with a connective layer, tissue layer underneath. They have some internal elastic membrane, particularly in the arteries. So there are elastic fibers in the tunica intima in the artery, so you can see some elastic membrane and more there in the tunica intima. So this would be the tunica intima, this would be the media, and then this would be the externa. So in the media we have smooth muscle cells, which are going to enable that artery to constrict or dilate as the case might be. We also have elastic fibers in there. And then the tunica externa is made out of collagen and elastic fibers, so it is connective tissue. Notice how the walls of the artery have more, I need to change my color there. The walls of the artery have more muscle than the walls of a vein. The blood pressure is not nearly so high in a vein, so we need more support in the artery than we do in the vein. Now here's more detail on the artery, the vein. So you can see a thinner layer of smooth muscle. Here's the endothelium, that's pretty much the same. And then here's the tunica externa. So what would be the difference between arteries and veins is arteries have thicker walls and they have a higher blood pressure. A constricted artery has a small round lumen. A lumen is just the opening inside a tube there. So we have that smaller, rounder lumen. A vein has a large kind of irregular lumen. The endothelium of a constricted artery is kind of folded, and arteries are more elastic than veins, And also some of the arteries have valves to prevent the backflow of blood. Arteries, since they have this extensive muscle layer in there, they are capable of vasoconstriction, so those muscles will contract in order to change the diameter, and that's going to be controlled by the sympathetic division of the autonomic nervous system. So vasoconstriction is when those muscles contract and reduces the diameter of the artery. Vasodilation is when those muscles relax and that enlarges the lumen. So vasoconstriction and dilation will affect the afterload on the heart, the peripheral blood vessels, and how the capillaries flow. The arteries from heart to the capillaries, arteries change. So first we have elastic arteries. So for example, the aorta is an elastic artery. It has to be able to expand and relax as the heart pumps. Then we have muscular arteries with thicker walls, and then we have arterioles, which are the smallest layers. The elastic arteries are also called conducting arteries. So these are going to be the large vessels, the pulmonary trunk, and the aorta. In these, the tunica media has many elastic fibers and few muscle cells. The elasticity evens out the force of the pulse. So it's very important for helping to control blood pressure. The muscular arteries are often called distribution arteries. Most of these are medium-sized, and the tunica media has many muscle cells. In the arterioles, we have what we might call resistance vessels. They are very small. They have little or no tunica externa and have a very thin or incomplete tunica media. An aneurysm would be a weak place in an arterial wall, which could be caused by a weak spot in the elastic fibers. In the worst case, that would rupture, and that could cause a lot of damage depending on where it is. It could cause death. Say if it was in the aorta and we had a rupture, it would be a very bad thing. So here we can see a diagram, and if we move to the next one, we'll see a little more detail. So here are the veins, so the larger veins. So we're going back toward the heart with these, and notice how they're getting bigger in diameter, and some change. These would be the capillary beds right here. And then we get smaller with the arteries, and again we can see the capillary bed. So arterioles and venules would be along there. Notice the particular structure of this arteriole with smooth muscle bands that don't go all the way around. And here we have a look at the capillaries. Now, there are different kinds of capillaries. This one is called a fenestrated capillary because it has all these little holes in them, are fenestra. So it's a very important filtering apparatus. Then we have a continuous capillary. Notice we don't have all those little holes. So a lot of the blood would just go through there and not lose much material. We also have a structure called a sinusoid, which we will take a look at. So the capillaries are the smallest vessels with very thin walls, and these microscopic capillary networks penetrate all of your active tissue. So anywhere you need a blood supply, you've got these capillaries. So again, if you removed all the other tissues from the body, you would still see the outline in capillaries and the other vessels. The capillary is the location where we exchange materials between the blood and the cells. So materials can move out of the blood and into this, eventually into the cells. First, you've got the capillary, and the materials move into the interstitial fluid, and then they can go into a cell. Conversely, we have waste materials that will go the other direction. So waste would include carbon dioxide and metabolic waste, and then the things we need would include oxygen and other types of materials such as sugars. So we just have a tube of endothelium inside a thin basement membrane for the capillary. We don't have any tunica media, and we don't have any tunica externa. Now the diameter is pretty similar to that of a red blood cell, or sometimes even smaller. The red blood cells have to often bend and twist to get through those capillaries. The continuous capillaries are found in all tissues except epithelia and cartilage, and they have a complete endothelial lining, so they don't have the little holes, the fenestra, but they do permit diffusion of water, small solutes, dissolved materials, and also lipid-soluble materials. Now they'll block blood cells, and they'll block the plasma proteins to keep them in the blood. We have specialized continuous capillaries in the central nervous system and the thymus that have very restricted permeability. An example of that would be the blood-brain barrier. The fenestrated capillaries have all those little pores, and they permit the rapid exchange of water and larger solutes. They're found in the choroid plexus, in endocrine organisms, in the kidneys, and also in the intestinal tract. The sinusoids are specialized capillaries that are found in places like the liver, the spleen, the bone marrow, and some of the endocrine organisms. They have gaps between the adjacent cells, so they sort of have little cracks in there, and they permit the pretty free exchange of water and even large plasma proteins. Now remember, the liver makes most of these plasma proteins, so we have to have a way to get this into the blood. So their sinusoids are found in the liver. There are also phagocytic cells around these sinusoids, which monitor the blood and try to gobble up some of the bad stuff. So here would be an example, fenestrated right there. Continuous here, you can see the fenestrated. And then here's the sinusoid. So these are the endothelial cells. And notice the little cracks in there, which allow for a freer exchange of material. And we have a blow-up. So this is a continuous capillary. So notice with no little holes. So materials that are lipid soluble, for example, can move in either direction, but other things might have a hard time. Blood cells can't escape. So here's the fenestra, the fenestrated capillaries. So it's kind of like a slit sieve, but those are still very small openings, so plasma proteins and blood cells cannot go between those. And then the sinusoid with the gaps. So let's look at a capillary bed. Excuse me. All right, so here's the arterial blood coming in, and we always show that as red, and the venous blood we show as blue. So this is deoxygenated blood, and this is oxygenated blood. This arterial, we can have constriction and dilation to let the needed amount of blood through. Notice here we have kind of a thoroughfare, and that's a continuous capillary. And then these other places where we get exchange, we can have fenestrated capillaries. And then here's another junction between the arteries and the veins. So that's an anastomosis, where those have joined. So if we don't need any blood in this area, we can shunt the blood around or through. And here's a photomicrograph showing you an arteriole, a small artery, and then the capillary. And just look at all those teeny tiny capillaries that are found in that area. So lots of blood going to that area. The collateral branches are those anastomoses that can directly connect an arteriole with a venule, and we don't have any exchange of materials. We have sphincters which can open and close to allow a certain amount of blood to flow through. Angiogenesis refers to the development of new blood vessels. So we have essentially a hormone, vascular endothelial growth factor. It's abbreviated there. And as organs develop, the body will secrete this material, and that will cause blood vessels to go into those areas. Also, if a tissue becomes kind of hypoxic or oxygen starved, then we may get the secretion of this material, and more blood vessels may grow into that area. Unfortunately, cancer cells can also produce factors which will cause blood vessels to grow to the tumor, and that will speed up the growth rate of the tumor once it starts receiving oxygen and nutrients. Okay, veins collect the blood from the capillaries and return it to the hearts. The heart, compared to the arteries, the veins would have a larger diameter, a thinner wall, and a lower blood pressure. Venules are the small structures that actually connect to the veins. So venules then go into kind of medium-sized veins with the tuna media and only a few muscle cells in that tunica media. The tuna externa contains a lot of elastic fibers. The larger veins have all three layers, a fairly thick tunica externa, a fairly thin tunica media, and some of the veins, especially in the legs, have valves in order to prevent blood from flowing backwards. This is especially true in the legs, so those can sometimes give you problems there. Varicose veins would be, those valves have weakened. Hemorrhoids can also result from the weakening there. So here's an example of some valves in the leg. So these are bicuspid valves. They have two flaps. So when the pressure here is greater than the pressure here, this valve will open and blood will go through, and then they'll close, and that'll prevent blood from going backwards. Now remember, the heart doesn't have so much of an effect on the veins, so we need some help here. So the contraction of these muscles is important. Also, veins are usually next to arteries, and the pulsation of the arteries can help. And then you have your respiratory system. So when you're breathing, during part of the period that you're breathing, you create a partial vacuum in the thoracic cavity, which also allows blood to move up more easily toward the heart. So here we have the distribution of the blood, and you can see only about 30 to 35% of it is here, and most of it is in the venous system. particularly these large networks here. Now, if you need that blood, then it'll be quickly pushed back to the heart and start going through the system to supply your needs. Here you can also see the distribution, and this looks a little bit strange, but notice that the heart only has about 70%, and then these structures have most of it. Veins can stretch more than arteries, so that's why they can act as reservoirs of blood. And systemic veins can constrict in response to blood loss. And that will help maintain blood pressure. So when you measure blood pressure, you measure it in the systemic system, not the arterial system. Now, the total capillary blood flow is equal to the cardiac output. And you have two factors here, the pressure, and then you have the resistance to flow. So pressure is that generated by the heart, which is necessary to overcome the resistance to flow. So we need a gradient of pressure. So we need a place where there's high pressure and a place where there's low pressure in order to get the flow of blood. And that resistance to flow increases as those blood vessels get smaller. So when we measure blood pressure, we're measuring the arterial blood pressure in millimeters of mercury, but there's other kinds of pressure that could be measured. The capillary hydrostatic pressure, the venous pressure is always low, but that could be measured too. The circulatory pressure is necessary to overcome the total amount of peripheral resistance of the entire system. So the change in pressure, that little delta means change in pressure, across the systemic circuit is about 85 millimeters of mercury. Now your resistance is affected by the resistance in the blood vessels, the viscosity of the blood, and the turbulent flow that you might find in blood vessels. Vascular resistance increases, notice, exponentially as vessel diameter decreases. So the smaller the vessel, the greater the resistance. The blood viscosity is just simply the kind of the thickness of the blood, and that's an important factor. If you have too many cells in there, it could become thicker and harder to pump, and then the turbulence is a factor also. Here you can see those shown. So a bigger versus a smaller, the change in resistance. And then down here we can see the flow. So here we have some plaque, some atherosclerosis, and notice how that's increased the turbulence. Now you want this nice flow like you see up here. That's called laminar flow, and the walls of that blood vessel are nice and smooth. See how we've changed from 1 to 16 with a decrease in diameter, and turbulence will also affect that. So those pressures are due to the diameter of the lumen, that's important. The total cross-sectional area, that's important. The pressures, and then the velocity of the blood flow. You can see, starting at the aorta, the diameter, so they get smaller, 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 and then they begin to get bigger, bigger, bigger as you head back toward the heart, with the largest being the vena cava. Here's the cross-sectional area, so notice the smallest at the aorta. You only normally have about 2.5 centimeters, about an inch in diameter there. And you got lots and lots of capillaries, so their total cross-sectional area is large, and then you get smaller again at the vena cava. And then here's the blood pressure, Now notice we need a gradient, so we need a place where there's high and a place where there's low. So it's nearly zero by the time you get back to the right atrium. Now this is not showing you that the blood pressure is actually going up and down, because we have systolic and diastolic pressures as the heart beats. Here's the velocity. So the greatest velocity would be coming out of the aorta. The slowest would be through the capillaries. And it's still slow but increasing as we move back toward the heart. So these diagrams are very, very illustrative. So we know we have two pressures. We have the systolic pressure when the heart is contracting, and the diastolic pressure when it's relaxing. And systole is our contraction, and diastole is our relaxation. So normally you might say the normal blood pressure would be something like this. These are millimeters of mercury. The pulse pressure is the difference between the two. So in this case, that would be 40. Now if you take the diastolic pressure and 1/3 of the pulse pressure, then you get what's called the mean arterial pressure, the MAP. When we get up into this range, say 140/90, we're talking about hypertension. Hypotension would be abnormally low. So, we like to think of 120/80 as being the normal, but there are people that have lower blood pressures and they are still normal. But on the high side, we like for it not to get over 140. If the diastolic is high, that might indicate atherosclerosis. Elastic rebound is just the ability of the arterial walls to stretch and snap back after systole. So they rebound, they recoil to the original shape. And that keeps the blood moving during the diastolic phase, so that's very important. The pressures in the muscular arteries and the arterioles, again, decrease with distance from the heart, and the MAP also declines as arterial branches become smaller and more numerous, and the pulse pressure decreases due to the elastic rebound. Now this shows you the pulse pressure, so you can see the rise and fall with systole and diastole, and then the red line representing the MAP, the mean arterial pressure. The venous pressure, again, is low. It's important, though, because it determines the amount of blood arriving at the right atrium each minute, the venous return. So the return to the heart is assisted, as I said, by the respiratory pump, and also the leg muscles are moving, and there's kind of a little pump in your foot when you're walking that helps to pump that blood back. Capillary exchange. So this is where the action is, where the materials in the blood can exchange with the tissues. And we need three processes here. We need diffusion, filtration, and reabsorption for this capillary exchange. So diffusion, we know we have movement from high concentration to low concentration. Net movement. There's also things moving from low to high, but the high to low outnumbers that movement, so we call it net movement along the concentration gradient. So things that can diffuse include water and ions and just small molecules. Sodium, potassium, calcium chloride are examples there. They can diffuse through channels in the plasma membrane. Large water-soluble lipid compounds can do that. We can pass through the fenestrated capillaries, so lipids and oxygen, and you can diffuse straight through the cell. Plasma proteins can go through the endothelium of the sinusoids. The filtration is driven by the hydrostatic pressure. That's just the fluid pressure, and that forces water and small solutes through the capillary wall. Larger stuff stays behind, like plasma proteins, for example. The reabsorption is a result of osmosis. So that's the movement of water through a semi-permeable membrane. The colloids, the plasma proteins, and other substances create this blood colloidal osmotic pressure. And those plasma proteins, remember, they stay in the blood. So here's the movement. Some small solutes are passing through here and getting through the membrane. So we have the interplay between filtration and reabsorption. That ensures that plasma and interstitial fluids are in constant communication and mutual exchange. It accelerates the distribution of nutrients, hormones. It helps transport insoluble lipids and tissue proteins. Also, it carries bacterial toxins and other chemicals to the lymphatic tissues and organs, which will help stimulate the immune system. So we have the net capillary hydrostatic pressure and the net capillary osmotic pressure of importance here. So factors that contribute to the net capillary hydrostatic pressure and the interstitial fluid hydrostatic pressure are very important, but this tends to push water and solutes out of the capillaries and into the interstitial fluid. Now let's think of a blood vessel here, and so this is the artery end, and this is the venous, and this is a capillary. So we tend to force materials out here at the artery end of the capillary bed, and then at the venous end we tend to reabsorb some materials. So the net capillary colloidal osmotic pressure is the difference between the blood colloidal osmotic pressure and the interstitial fluid colloidal osmotic pressure, and that's going to pull water and solutes into the capillary from the interstitial fluid. So we can figure all these up and get the net filtration pressure, and notice we have this minus this minus this minus this, and that gives us the amount of pressure causing filtration. So we lose fluid at the arterial end and we gain fluid at the venous end, and right in the middle we're kind of equal filtration and reabsorption. Now the capillaries actually filter more fluid than they reabsorb, and if that continued to happen without any way to relieve that, then fluid would build up in the interstitial area. But we have the lymphatic system which will collect that fluid and return it to the circulatory system. So here's a diagram illustrating what's going on here, and notice your numbers. The net filtration pressure up here, so maybe we need black, up here is plus 10 millimeters of mercury. So you take this pressure and you subtract this other pressure. So this pressure is pushing fluid out, this pressure is absorbing it, but notice you got 35 minus 25, so we got 10 here, and so we filter 24 liters a day, lots of fluid, and at the center we're about equal, but at the end, the venous end, notice we've got a minus 70, So that means fluid is moving back in more than it's moving out. Now you want to notice the difference. Here we have 20.4 and here we have 24. So that's 4 liters a day that we're losing. Well, our old friend, the lymphatic system, picks that up and returns it to the circulatory system. Hemorrhaging reduces that colloidal hydrostatic pressure and the net filtration pressure because you're losing those colloids out of the blood, and that's going to move more fluid into the interstitial fluid. Dehydration will increase the blood colloidal osmotic pressure, and that will accelerate reabsorption. So if CHP or BCOP declines, then we'll get movement of fluid out of the blood and we'll get edema. Or if the lymphatic system is blocked, we can get edema. Now, perfusion refers to the amount of blood flowing through the tissues. So we want good perfusion so we can get the oxygen and the nutrients to the tissue, and we can get rid of the carbon dioxide and the waste materials. So all of these factors are going to affect the perfusion. If your cell becomes more active, then you need to pick up the circulation. So for example, if you're using your muscles, then you're going to have to get more blood to the muscles. And the cardiovascular system can do this. It can move the blood to the right place at the right time without changing blood flow to the vital organs. And particularly you want to think about the brain here. We need blood flow all the time to the brain. Vasomotion then just refers to the ability to constrict or relax these blood vessels in order to get blood to the right place at the right time. Mostly that's going to be under control of the precapillary sphincters. Now you can control all of this through several mechanisms. One of them is auto-regulation, so you can make local adjustments in the amount of blood going to an area. Then we have neural mechanisms to respond to that, as we will see. And then, of course, we have endocrine mechanisms which will respond to that, and these are more long-term, and the neural and the auto-regulation are short-term and happen quicker. So here's an example of the homeostasis. So notice we can see auto-regulation. So for some reason we've had a change here, a stimulus, and so we've had changes in the amount of oxygen or a change in the pH or the carbon dioxide or some other factor, and so we're getting inadequate blood flow there. The precapillary sphincters will respond by dilating, and that will decrease the resistance and increase the blood flow. Now if that doesn't work, then we have these factors come into play. So we have the nervous system, so we have changes as a result of stimulation of certain receptors there, and we can tell the center in the medulla, in the brainstem down there, to do some activities, the sympathetic nervous system, and we can increase the blood pressure and we can increase the volume by having an endocrine response. So that would be shorter term and longer term, vice versa. This would be pretty quick, and this would be a little bit slower, but last longer. So this autoregulation is very important to blood flow within the tissues. We've got vasodilators that can promote the dilation of those. So oxygen, carbon dioxide levels can do that. Lactate can do that. Nitric oxide, high concentrations of potassium or hydrogen. That would be a low pH. Histamine can do that, and even the temperature can do that. The cardiovascular center of the medulla oblongata is important. So we have cardiac centers to speed up and slow down the heart. And then we have vasomotor centers to control the contraction or relaxation of particular blood vessels. So the vasomotor center will control vasoconstriction or vasodilation. And that center is always putting out some impulses in order to maintain tone. So like your muscles are kind of partially contracted and have tone. The cardiovascular centers monitors the arterial blood, and we have baro and chemoreceptors. So the baroreceptors are sensitive to blood pressure changes, and then the chemoreceptors are sensitive to pH, carbon dioxide, and oxygen. And so these amount to reflex activity. So in your carotid sinus, now that's a kind of a widened area where the common carotid splits into the internal carotid, and this is in the internal carotid. You've got to make sure you get blood flow to the brain, and the internal carotid is doing that. And then the aortic sinus is kind of a wide place right after the aorta pops out of the left ventricle. The right atrium is monitoring that. So changes in blood pressure in the aorta trigger the aortic reflex, which adjusts blood pressure, and that's going to be for the systemic circuit, arterial blood pressure. So we can see that here. So the baroreceptors, which are sensitive to changes in blood pressure, are stimulated. They send a message to the medulla, and then we activate these centers as necessary to reduce the blood pressure in this case. So that's a protective mechanism to try to keep the blood pressure under control. Okay, here if the pressure is too low, then these receptors are inhibited. Once again, we're sending messages to the medulla, so we want to raise the blood pressure in this case. Okay, the carotid bodies and the aortic bodies are monitoring the chemicals here, chemoreceptors. We're also monitoring the cerebrospinal fluid there, and we're controlling respiratory function. And we want to make sure that blood is going to the brain. Remember, the brain needs a constant supply of oxygen. It needs to get rid of that CO2. Just a few minutes without oxygen, and you're going to die. And this is going to respond to changes in the pH also. So here's our mechanism here. We've got the stimulus. Stimulate the receptors, so we have increasing carbon dioxide and decreasing pH levels. So the system responds, and we will try to restore the blood flow to proper homeostasis. So we increase the blood flow, we increase the rate of respiration. Your emotions can affect blood pressure. You get scared or get excited or get sad, your cardiac function will change. Hormones they have some short-term effects, but then they have long-term effects. epinephrine and norepinephrine for your fight or flight, they'll get a pretty quick response. And then we have ADH, antidiuretic hormone, and that will elevate blood pressure, cause the kidneys to conserve water. So this ADH responds to low blood volume, which would have a low blood pressure, and that would mean you'd have a higher plasma osmotic concentration, and it would exert its effect through the angiotensin route. So when this angiotensin is produced, then we'll also get the production of aldosterone, more ADH secretion. We'll be thirsty. So if the renal blood pressure drops, we'll take action. The kidneys like to have a good high blood pressure, or they can't filter. If erythropoietin, EPO, is released by the kidneys, and it responds to low blood pressure or a low oxygen content, stimulates vasoconstriction,